Thank you very much, Dr. Campbell. And I'd like to also thank the organizers for what's been a very stimulating and interesting day so far. I'm here to talk to you today about the role of endocannabinoids. Let me just share my screen. And their potential utility of a Fa inhibitor or fatty acid amide hydrolase inhibitor in the treatment of autism spectrum disorder. I think we've probably covered by now as much background on autism spectrum disorder today as many of you may need to know or already do know. What I'll just mention before we get started is that there are, of course, a number of clinical targets. We hope that the treatments that we're looking for now target the core symptoms of autism spectrum disorder, that is social communication deficits and restricted repetitive behavioral excesses. But we also hope that the many comorbidities that are present in individuals with autism spectrum disorder can be improved with various therapeutic strategies. And until now, there are no approved medications for the core symptoms of autism, and we really need a much better armamentarium to treat the symptoms and to improve outcomes for individuals that are affected by autism. One thing that is, of course, very compelling, and I know has been discussed before as well today, is that genetic research is rapidly advancing our knowledge and our understanding of ASD neurobiology. And that has led to really an explosion of research in small biotech companies and in larger companies to try and understand the biological systems that underlie autism. So let's talk a bit about the endocannabinoid system. We know that the endocannabinoid system promotes emotional homeostasis. It mediates the environment and signals in the brain, particularly around the reward and stress systems. It modulates physiological function, including mood and anxiety, and there's considerable evidence from both animal and human studies to demonstrate that. It also has a widespread effect throughout the brain and in the neurotransmitter systems, including impact on dopamine projections to the limbic system, and also on stress system, including the hypothalamus and the amygdala. Endocannabinoid inhibition also has a potential antidepressant effect, reduces anxiety, that is, enhances the recall of fear extinction, and attenuates the stress response. It reduces stress, perhaps preventing stress effect of decreased anandamide and amygdala hypertrophy. And what I'll focus the majority of my talk on today is improvements seen in social behavior related to autism from animal models. And we note there that we see increases in prosocial behavior and decreases in repetitive behaviors. So I'm going to talk with you about various aspects of the neurobiology, and I'm happy to answer any questions at the end. So early evidence came from genetic and behavioral autism models that implicated the endocannabinoid system. And in this publication by Kruger and Bros, they summarize reports by Foldy looking at the impact of neuroligands facilitating synaptic transmission. And when we knock out neuroligin 1, 2, and 3, you can see dramatic reductions in transmission. And that also, in ASD-associated neuroligin 3 mutations, disrupts tonic signaling of the endocannabinoid system, which we know then also has an impact on glutamatergic signaling and AMPA. So this excitatory inhibitory imbalance is dramatically impacted by these pathways, and this relates also to the endocannabinoid system. So oxytocin also has an effect on socialization that's mediated through the endocannabinoid system. And here we see some preclinical data showing that oxytocin receptor activation enhances anandamide mobilization in the nucleus accumbens of the mouse. So oxytocin functions as a social reinforcement signal in this limbic region. You can see here in the model on the right. We also know that heightened anandamide signaling via inhibition of Fa stimulate social reward, and that occludes the prosocial effects of oxytocin. So these relationships between different hormonal systems and signaling systems in the brain are all different potential ways that we may be impacting on autism symptoms. Here we see some genetic and behavioral data that implicate the endocannabinoid system. And in the prenatal VPA exposure model of ASD, we can see that both MAGL and DAGL enzymes are related to endocannabinoid synthesis. So this is the VPA knockout model. And you notice that MAGL and DAGL are both deficient in the MAGL in the hippocampus and DAGL in the cerebellum. This suggests that these enzymes, which are related directly to the endocannabinoid system and catabolism, may have a significant role to play. The endocannabinoid system may also be important in social play in mediating novel social encounters, and there's considerable data that's been published about that. So we see some of the best preclinical evidence comes from the Fa inhibitor URB597. And in this model, we see that an increase in social play behavior in rodents and a decrease in repetitive behaviors, specifically head twitch response. And these are just two examples of that data. You can see other publications that I've noted here on the slide referring to preclinical models of reversal of ASD behavior under the condition of Fa inhibition. We also know that the endocannabinoid system plays a role in seizure activity. And in this particular slide, you note that, well, first of all, we know that seizure comorbidity is approximately 25 to 30% in the autism community, and a common problem, often intractable. And the onset of seizures can also be across the lifespan. So while you do see seizure onset early in childhood, you can also note that it will occur spontaneously later in adulthood. And so not only is it an intractable problem, but it may also be a neurobiologic symptom of a cascade of neurobiologic problems or electrical problems of the brain. So in this particular slide, you see that the Fa inhibition reduced EEG seizure activity in a PTE-induced seizure model. And this is important because part of what we continue to have challenges with, as was mentioned before, is heterogeneity. And of course, we know now a number of single gene disorders that can result in a very high comorbidity, sometimes even 100% with autism diagnosis. And these then, of course, are mirrored by our work in genetic research that suggests that copy number variations or large gene studies triangulate these genes is important for autism. So not only do we see them in single gene disorders, but we see them as risk genes. And what this suggests to me is that we're having a cascade of problems in the brain that could be formed in multiple ways with multiple different insults at multiple different time points, but that the final common pathway may result in these large effects, such as an increase in seizure disorder. Another aspect that's quite interesting, and these are now human data, actually post-mortem data. There's really some stellar research going on and really heroic efforts by researchers to learn as much as we can about autism through different mechanisms. So there are a couple of brain banks where individuals who with autism who pass away donate their brain. And in this particular study, they noted that decreased CB1 expression occurred in multiple cases of autism. In particular, you can see the gene bank ID here in the locus for those of you who are interested, but there was approximately 30% decreased expression compared to controls. The CB1 receptor, of course, is directly involved in and related to the endocannabinoid system, and this we think is an important marker for autism. Another line of research that relates to the biomarkers that Dr. Campbell mentioned, biomarker research that we're doing that Dr. Campbell mentioned, is looking at social gaze. So in particular, we know from a large body of research that eye gaze in autism, which is a big part of social engagement and social interaction, can be impaired. And in fact, looking at eyes and looking at faces, as I would be trying to do to you if we were talking in a real hall, but unfortunately we're doing this through the virtual space, can be a marker for social engagement. And we learn much about the social interaction and contact by eye gaze. And this was a study of typically developing individuals. And what they did, though, was to look at the impact of the cannabinoid receptor, the CB1 gene, and the influence of that on eye gaze. And so it's a simple task of viewing faces through a screen, how much eye gaze there actually is. And we can look at this either in dynamic ranges, so in video, or in static images, which are just static on the screen. And in this particular case, we look at the eye gaze trail on a still frame of an individual showing a happy expression. And the fixation points represent the amount of time spent. And what we noted was that for the groups that had the genotypes more closely linked to CB1 receptor deficit, the less likely they were to be looking at eyes. So this may even be a genetically determined in some instance function that may be we know to be variable in autism. We know that not all individuals with autism have the same level of difficulty or the same level of lack of attention to eyes and faces. So this is confirmatory data from individuals with a CB1 receptor gene. So this leads us back to that other flip side of the coin here. I want to just mention briefly, as I think others have talked about today, that there are other things that we know about the endocannabinoid system, including cannabidiol and THC. While there are certainly very limited data, there have been some studies here. It is not uncommon, for example, for medical marijuana to be prescribed in states where it is available, particularly for seizure control in autism, but there have been some symptomatic benefits that have been noted in case reports. By no means am I advocating this as a treatment for the core symptoms of autism, but we do know that things like cannabidiol are now under study, and cannabidivarin that are under study. And while some of these studies have been, the results of these studies to date have been mixed, I think there's more data will be emerging on this and the potential role of the endogenous cannabinoid system versus exogenous cannabinoids. So I wanted to end today by talking a little bit about our biosensor research, and I'm just going to give a couple of examples. We mentioned before that autism is a very heterogeneous disorder. And so over the past 8 years, we've been trying to develop tools and technologies that might help us to understand better autism spectrum disorder. And so we've developed a two-component system, one which is a mobile medical application called MyJake. And this is titled the Jannsen Autism Knowledge Engine. We had to pick a name in order to be able to launch an app in the App Store to be able to download for clinical trials. It's available only for research purposes at this point. And what we've done is try to implement different approaches to tracking of behavior. So we have smart rating scales, which are clinician or caregiver reported outcomes, which are the primary outcomes that we're looking at now. And we've been developing a scale called the Autism Behavior Inventory that we hope will provide better endpoints, particularly in the short term in clinical trials, as a measure of core and associated symptoms of autism. But we also think it's important to look at things like caregiver selected most important symptoms, other measures of mood and behavior, daily measures, and also other ways to assess behavior as opposed to standard ways, because we know that autism can be heterogeneous, and we don't want to miss symptoms or subtle changes, particularly if we have subgroups or subpopulations of individuals who may be responsive to medications or combination treatments. In addition to that, we've been developing what we call a biosensor workbench, which we call Jake Sense. This has a series of continuous biosensors that are used at home, including actigraphy and others. We also use biosensors in the lab focused on different paradigms, so social versus non-social videos or static images, brain-related EEG functioning, resting state, and ERP, and other types of tasks like this. And we look at, beyond just eye tracking and EEG or electroencephalogram, we look at electrocardiogram, where there's some very interesting data that we're analyzing right now, and also facial affect data, which we've published on before. There's a whole large community of researchers looking at these types of endpoints now. There's a consortium in the United States called the Autism Biomarkers Consortium for Clinical Trials, and there's also a large consortium in Europe, the AIMS2 Trials Consortium. And I encourage you to go and visit their websites to look at the research that they're doing to better understand how these biomarkers may be helpful in identifying more objective behavioral measures and things that might allow us to subtype or stratify, and also to measure change in individuals with autism spectrum disorder. This kind of research, in complement with genetic research, metabolomics, and other emerging fields, we think is going to be instrumental in better understanding autism and helping us to develop better therapies. So in summary, I hope that I've given you a bit more information than you had at the beginning about the endocannabinoid system. It's implicated in the modulation of mood, anxiety, and stress. It also has a distinct role in the modulation of social behavior. And there's evidence from preclinical models that demonstrate reversal of social deficits and repetitive behaviors with inhibition of Pha. And lastly, CB1 receptor have been demonstrated to be downregulated in autism, and that those CB1 receptors are implicated in social behavior as well. In the interest of time, I'll stop here, and I'm happy to take any questions that you might have.
Thank you, Dr. Saunders, or Dr. Pandina. That was great. So I did receive a couple of questions here. There's a few coming in. You covered a few different topics there, and so they'll be a little bit diverse in questions. Regarding the eye gaze tools that you referenced, have you considered using these in populations in which the phenotype is more consistent, such as Rett syndrome, for example?
There are researchers that are doing that right now. We've been taking an all-comers approach, in particular because as we develop them, we want to know what they might be sensitive to, and looking at the behavioral phenotype. What's been interesting, though, is that there's only been a modest correlation between deficits in eye gaze and symptoms on symptom rating scales. Now that could be because the symptom rating scales we have are only measuring certain aspects of behavior, but likely that's also a heterogeneous finding as well.
Okay, and related to that, I apologize, I don't understand the acronym, but have you looked at HRV as one of the biomarkers for tracking via sensors?
Yeah, so heart rate variability, I think is what's being referred to there, is an interesting concept. It's an interesting finding. We see that this relates to stress. We know it relates to, it could be a marker for stress, which could, and social stress. We have some data from our both our at-home measurements and the lab measurements that suggest that there are differences between individuals across the board in heart rate variability and other heart rate and other variables that are important. And we'll be looking at those in the coming year in particular, and hopefully we'll have some publications next year talking about that data. But it's a very interesting area, in particular because cardiovascular stress is something we know individuals with autism suffer from, and we know from the UK reports that they're very that unfortunately life is foreshortened in autism, often not because of the autism itself, but because of these comorbid conditions.
Mm-hmm. And I guess, would it be of interest or of any potential value to look at HRV while somebody is being assessed in a behavioral assessment?
Absolutely. What you'd want to know, so we have a couple of paradigms where we've done this, where we've actually introduced a tone or an unpleasant sound, not at a level that would be so distressing that it would sort of derail the research.
Sure,
You can
sure.
imagine that you could do that.
Mhm.
But these, and it's interesting. We will be looking at those data in more detail. It is challenging to get those types of data, but those are kinds of things that really do lend themselves to both in. What you need to know, though, is the context, the direct context.
Exactly.
Just like an EEG, when you're doing an ERP research, you have a stimulus that's presented, and then you have to know exactly when it was presented and what happened thereafter.
Yeah,
So,
I guess it's akin conceptually to fMRI, right? We have very tight control over the tasks being asked and demanded, and you see the response in the imaging, right?
yeah, people have tried to use other things like electrodermal reactivity, so skin conductance as a measure of stress, similarly, and to follow people and to tag behaviors while that's happening. It's a little grosser measure because it follows behavior by a little bit longer, but that's also being used in seizure detection and other things. So we do think these physiological measures are going to be important as signals of changing biological states and hyperreactivity in autism.
Yeah, and I guess the reason I mentioned Kevin Saunders' name was he alluded to this big data problem, and I was thinking as you were going through this, you collect all this stuff through Jake, and then you have to integrate it somehow. You have to find a way to analyze it in a meaningful way. And so what's the back end of this look like for you folks?
So what we really do is we create a series of small experiments. So we have a series of challenge tasks. We pick specific variables up front that we think are going to be important and relevant, and we make predictions about how they should be different directionally in autism versus typically developing controls, and then we compare them to a normative control group. So that's one approach. That's the sort of standard, approach that we use. But we also think that machine learning has a role to play here in automated analysis. And what we'll do is put everything in the soup, do recursive analyses to remove redundancies, and then when we do that, and I've given talks on this before, we often end up in the same place. So we end up with there's some variables that seem to really predict whether or not someone has autism or not. And those variables seem to arise from the machine learning data as well. Of course, these are always, it is challenging when you have a very heterogeneous population. And what we wanted to do is to create a system that would also allow us to assess the tails. So people with individuals with autism who were had normal or above average IQ and generally good communicative ability, to people who are almost nonverbal and unable to communicate or engage and may have behavioral problems.
Yeah, no, that makes sense. Final question here. Have you looked at patients that have a history of a fever response as a potential subgroup? They're noting that activation of CB1 has been shown to induce fever, so they're curious if responders may have a history of being a fever responder.
That's interesting. We have in our behavioral data tried to collect, or in our history data tried to collect information on fever response, because we do know that there are individuals who report that in their history as being something that was developmentally traumatic, both probably for the parents and for the individual with autism. We haven't looked at it in our data. We don't have a robust enough data set to do that, but it's certainly something that we as a field should pay attention to, other biological states or hyperreactivities.